we might get some idea of this from the industrial data sets that we get in.
Mm-hmm.
But
Yep.
we discussed other subgroup analysis in Florence last year. So, for example, you know, the genomic class that we termed
Yep.
the spliceosome group. I think this is still a rather heterogeneous group, and to use this large data set further to further characterize this group, I think this is worthwhile, and probably the same is true for the other that we call the TP53 chromosomal aneuploidy. That this is also quite heterogeneous.
Mm-hmm.
To specifically look at these two genomic classes as we defined them in our paper, and to use this data set, you know, perhaps to further subdivide or classify these cases.
Mm-hmm. Yeah, I forgot to put this on the list. That's correct. But this is also something which we easily could do based on the data that is already in there.
Yeah.
Yeah. So this is Gastone. You are also in the, yes, you are in the call. So this is something which
No, I'm here, Lars. If you want, I can give some, let's say, technical or update on the machine learning or the algorithm we have. So if you want, otherwise your biological medical discussion is very interesting. I will learn from you.
No, I'm not sure whether, yeah, I think whether this is helpful. I mean, but more or less the platform is set up in a way that all the people that were interested in participating in the data analysis, they were getting access to the data set. So I think also the Rotterdam group could access it. People from Helsinki could access it. And the platform is also constructed in a way that you can run your own R scripts. Okay.
Yeah. The problem is just the interface with OMOP. At my college now, only 3 groups are using this interfacing with OMOP. Your group, Eric Strand, the group from Salamanca with Javier, and the Bologna group. So if other group want to analyze this data, you will need to know how to interface with OMOP, and we can help you, but it's not trivial. It's enough complex.
Mm-hmm. But I think for groups that are well versed bioinformatics people, I think that should be doable, right? So like Caroline Heckmann's group, or for example Moritz Gerstung's group, or the guys from Rotterdam, I think they should be able to do it.
Absolutely, yes, but it's not trivial. It's will require some effort and some dedicated people.
Yeah.
Because OMOP is not intuitive. This common data model is wonderful because it's a common data model, but is not trivial. So I don't know if there is some other that would want to add some, but it's not trivial. And also in the platform we have some limitation. For example, I have some algorithm written in C, and in the platform we can't use the command line. And so now we are reconverting this code in Python. So there are some technical difficulties, but of course the system is not open, but it's possible to access to other people. And
yeah, but I mean, then I think the straightforward practical way to approach the data then at the moment would be that you have defined research questions, such as the one that Hartmut now just pointed out and which we had discussed already last year. That's like that we look at these specific genetically defined groups with spliceosome mutations or with P53 mutations and chromosomal aneuploidy, and that we then perform the analyses. it gets a little more complicated than if we want to perform these models that Moritz Gastung has implemented. As I think they are not in there yet, but I think I have already shared the paper with you, Gastone, right? And Javier in Salamanca is working with similar models to come up with these multivariable analyses for these gene-gene interactions. And I think
from a practical point of view, Lars, so who will do these analysis? So just the project I mentioned. So from a practical point of view, are we doing these analysis? Are you doing these analysis? How does it work on a practical basis?
Yeah, this is what I just wanted. Yeah, I think this is what we have to discuss. I mean, so far on a practical basis, only those 3 groups can access the data and work with it. If you have, for example, people who want to do the analyses, for example, like Axel Benner or so, I mean, then we can include him in the data analysis group. Then he would get.
I was about to say the same.
Yes.
I was about to say the same thing.
Yeah. Hello, Caroline. Then they can access the data and run the analyses. Or the other way would be that he tells us what he wants us to run for him on the platform. And then we share the results, and then we sit together and discuss the findings, and then go from there for the next round of analysis. But Caroline, sorry, what did you want to say?
Okay. You muted, Caroline.
Okay, no more Caroline.
So can I comment? This is Kimmo from Helsinki.
Yeah.
So just a comment on what Gaston was saying too. So at least we are facing more and more challenges in transferring patient level data out of the hospital databases, including some of the trials data. But what we are doing currently, so we are transferring all our data sets at the hospital side to the same data model now, to the OMOD data model. OMOD data model, and I'm wondering that whether it would be possible to do these analyses in a more federated fashion, that you would just circulate the code and not transfer the actual patient cases, because that way we have maybe 1,500 patients ready to go in the same data model, but it will be quite challenging today to transfer these data, just this hospital data to the Harmony database.
Yeah, sorry. Now I'm back.
Okay, well, please.
Yeah, so what I was going to say earlier is actually there's an SME attached to Harmony Metasapiens, and actually what they do is homopization of clinical data, and I think it would be actually relatively easy for them to probably just set up the workflow to do this for most of the data sets that are coming in.
Yeah, thanks, Caroline. No, actually, in the platform, there are two notebooks in Python, and by using this notebook, you can be interfaced to AML data or to multiple myeloma data. And so if you want, you can use this interface just to query the AML data and to run your analysis. So this is, no, actually they are in R, not in Python. But I think in the next week it will be great, and I will organize a meeting for the guys that want to run the analysis, because this will be very, very useful.
Yeah, I think this is too technical for most people in the audience here, Gastone.
Yeah.
But nevertheless, there is the possibility that if you have people at your institution who want to be part of the data analysis team, that they can be part of it. But I think what would make sense, I can perhaps then send around some minutes that we agree that we start to do the analysis with the people that are already involved in analyzing the data, and that we assign for each of the individual projects also a PI who is responsible for then looking at the data and coming up with additional ideas what should be analyzed next. And I think this is also what we need, right? Then, because I mean, then Gastone and Javier and Eric, they are producing a lot of data or results, but then we also need someone to look at the results and to make sense out of the results from like a biological view or from the clinician view, right? As you had mentioned, for example, we have these nice curves, they separate, but then of course, then Hartmut's point is very valid. We need a multivariable model, for example, and then we should sit together and discuss which multivariable model we should use and what should be entered. And then you can again go to the next level, do these multivariate analyses. And as soon as we have the results, we have again a joint meeting. The question is whether we do that for all the AML projects always together, or whether we should form smaller groups and indicate perhaps then also young people who are responsible of one of the respective projects who also have time to do so, right? Because to always get this large crowd of people together is, I think, almost impossible more than twice a year or so. But how should we do that? And there I'm really open for ideas. I mean, I think definitely Hartmut, you would like to be involved and have the lead on the spliceosome group and P53 chromosomal and ploidy group.
Yeah, I mean, yeah, we can do it. I mean, we can do it as a team. I'm open to, to solutions,
Okay.
But yeah, that's certainly something that we are interested in. And as you mentioned, I mean, we could involve those people who have been working on that, like Axel, if they can be part of the analysis team. I think that would be interesting from our side, because we have entered quite a lot of data.
Yeah. And then also, I mean, this is someone who knows the AML projects, which we have performed very well. He knows, he brings in a lot of expertise. Yeah, and I think for the MRD project, definitely, I think Hertz Group would like to be involved. And I think with regard to Peter, you also had some aspects where you wanted to take the lead, or do you have any ideas what you would like to investigate in more detail?
Well, AML MRD is of course definitely of interest, but I can imagine that may. So obviously you have flow and you have NGS data there.
But I think that the MRD proposal had proposed, I think there we still have to collect the data sets.
Yeah, exactly. So that was exactly my point. So the flow data, well, is maybe a little bit more mature because it's simply already there for some more time. The NGS data, and especially the standardization or harmonization of the data from different centers, is probably even more challenging than having the data at diagnosis. So obviously it would be very interesting to have these numbers of cases and also MRD data. But well, obviously the data is not yet there. And I mean, we are of course working hard in the ELN MRD working group. So maybe there's also a link there where we could team up and see whether we at least do something similar if we start doing NGS MRD, such that the data sets, well, maybe not next year, but in the years to come will be, let's say, harmonized. But I mean, obviously, Amsterdam and Rotterdam would like to be involved in the MRD analysis in AML in harmony setting. Absolutely.
Good. Okay. But any specific genes or subgroup analyses or specific mutations you would be interested in?
No, well, that's also something to really think a little bit harder, but it's not so simple. I think because it's, of course, mutational data of a subset of genes, and obviously there has already been done quite a bit in the past. So you do not really want to be only confirmatory, but really find new things. And even as you have showed now, even having like 5,000 cases, it is still challenging to really study the smaller groups. So I mean, this is something to think a little bit more about how to approach this. So I do remember that in Florence, Christian also suggested the CBP alpha
Oh yeah, that's
analysis.
true. I forgot
So
that
I
too.
think you should also add that. So that would definitely also be relevant. And one other thing that I may have brought up in Florence already is, so the germline analysis, which is of course not really, I think, part of this initiative, but that may, I mean, that may definitely be of interest, especially, or at least of some of the mutations of which we know that there could be germline involvement, like CBP alpha, but
based on the varying allele frequencies, perhaps we can get some clues. That would be also something to mine the data with that regard.
It's of course not so simple because you do not have normal tissues, of course, but maybe there's something there.
Okay, good. Okay, but what would be the concrete next steps? I'm really, this is where also really looking forward to your input. I mean, one thing is, of course, we can now do some analyses for these points, share the data with all of you, and then we have another work package AML meeting in, let's say, 6 weeks or so, and discuss the data. Would that make sense with the people interested in the respective questions, or should we then do it in smaller groups first and address, for example, for the, let's say, spliceosome P53 chromosomal aberrations, first talk to Hartmut, what he thinks, get his input, refine it a little further, do some more things, and then come with more concrete results to everybody? Or would everybody like to be rather involved right away from the beginning in more or less the first round of analyses? The problem is then, of course, always, I mean, everybody is spammed with stuff, and then if no one is responding, that, it's always, I mean,
this maybe also depends a little bit on whether the goal as a first deliverable would be to have a high-level overview paper on the 8,000 patients. And then the focus of that, I'm not really clear yet. I mean, you showed the gene-gene interactions and things like that, but should the first deliverable be like an overview of the Harmony data set, or should it be several specific questions like the one we discussed, BTPN11, spliceosome, and so on? In the first case, probably it would be good if you take the lead with a small group and you come up with an overview of what could be done, and then there's a meeting in the big group. If rather the second, several smaller project is the aim, then probably it would be better to define leaders for each work package and have them work on it individually.
Yeah, so I mean, the plan would be to come up with this overview and this huge gene-gene interaction study, perhaps a little similar to what we have done in the New England Journal with these 1,500 cases. Of course, this makes only sense if we also then find something new, and if it's not only, I mean, not only confirmatory, I agree. But then during that analysis, I would assume that we already get a glimpse of what interesting subgroup analyses might be worth also to be further pursued, and then we could assign those to individual or PIs. Yeah, that perhaps that's a good idea. But then we would proceed with the overall analysis and share it with all of you. And for the subgroup, but then the question is, of course, again, then if we, let's say you are interested in looking at CBP alpha, would you then rather like to be involved with your own team, or should the teams that are part of the data analysis group at the moment rather do the analyses and share the results? This is also something perhaps makes sense to, I mean, or if everyone can send me back a notice whether their group has people who would like to be part of the data analysis group, such as Axel Benner, then, for example, I would approach him now for the Ulm group and say, would you like to participate in the data analysis for this overview and the gene-gene interaction on the multivariable analyses? And then we can put him in here, and then we have a group of people working on individual questions. I think that makes sense. Was this clear to everybody? Perhaps not, huh? But I think I think everybody who has a bioinformatics person who would like to be part of the data analysis, I mean, they could name the person to us and we make sure we involve them. And then perhaps it would make sense to have a meeting with the people that can access the data and analyze the data and assign tasks to them. And then we share the results with you. And then we go from there. Okay. No one is, no votes against that part. Yeah, okay. I think that we move forward with like that. But then now it's mid of October. I think we should share something with you by the end of November. Because then we also have to prepare the presentation for ASH, and there we really want to show new stuff and not what I've shown you today. Good. And perhaps then, I mean, we should not forget that, Maria, you also have to do a presentation. Hartmut has to leave at 5. Or do you have 5 more minutes to look at the slides?
I have another meeting at 5. I have to leave. I'm sorry.
Sorry.
Yeah. So
I talked too long.
Bye-bye.
Bye.
Bye, Hartmut.
Okay, does everyone agree with, and it's hard with this virtual meeting, so there's no objections with regard to how to move forward like that? Or Brian, do you have a better idea?
No, I mean, I think that it's exactly the way to do. I think as we've sort of discussed in Florence and as we've sort of reiterated again today, I think there are obvious areas that people are interested in that we can already see. And I think we should start to put together those teams. You're absolutely right, particularly when you get the larger numbers, it will give a lot more granularity. And I think that new questions will start to arrive. So it's difficult to second guess those in many ways. Yep. So I think that this is probably the most sensible thing to do, rather than chasing shadows. You know, to get the data in, get some preliminary analysis done, and then the size will start to open up new questions, and then it's a case of, just as we've done today, trying to work out whether it's the big group that tries to tackle that, or I think my preference would be that it's smaller and more focused groups.
Yeah. Yeah, the problem is always a little bit, we talk a lot. I mean, as you mentioned, we discussed more or less the same things last year. Yeah. But then you put together the people, and we have a large round of people that want to contribute. And at the end, then due to technical reasons, whatever, it's always few people who do something, right? But I think to be really successful now in the final year, we have, yeah, perhaps to identify really people who want to do it and assign a task to them, and then they have to do it and share the results with the others.
Yeah.
Yeah.
Yeah, what you mentioned, to have some of the people who are, one is responsible, 1 or 2 for each sub-project, and then they will take the lead. That's the way to the projects, the sub-projects.
Yeah. Yeah, but I think we need also young people who have time and are dedicated, and then on the analysis side the same, right?
Yeah.
Yeah. Okay, good. But then that opens the floor for your presentation.
Hey, Lars, can I make one comment? So this is Peter again. So how difficult would it be for you to redo the analysis you did in the New England Journal, so with Ellie, because that is then sort of a reference.
Yeah.
If you would, I mean, obviously it would be in a way confirmatory, but what you at least hope is that you will find things on top of what you have seen then. And that may in fact open new projects. And then if we look at it together, then people may also be interested in some of those and want to really pursue those. Because this is, I mean, it's obviously a quite complex data set, of course. So it's quite hard to say, well, I would like to focus only on CBP alpha, but at this point, I do not know whether there's anything in CBP alpha, or I would like to focus on one of the splice factor mutations. But at this point, we do not know if there's anything really new in there to decide at this point, okay, I would be interested in this. And that's, so I was just wondering if you would be able to do this, redo this analysis, or someone who was involved in this analysis, then you suddenly may see things that could potentially be of interest of people who are interested in a particular mutation anyway.
Yeah, but this is more or less, I think, what goes in line with what Hartmut suggested, right? If we have a deeper look at these two newly defined subgroups, which we...
Yeah, but maybe not only those. Maybe
so,
just
I mean,
the
we
data
could
set as a
redo
whole.
the analysis on the larger data set and see whether we still see the same grouping, or whether you see new subgroups and whether they hold true. And the other approach would be specifically look for those subgroups, how they behave in the larger cohort. That would be confirmatory. Yeah, we should do that. I mean, this is also why we wanted to have Moritz Gastung in the data analysis team, and he also said that he would participate. I think he's now moving from Britain to Heidelberg. I had did I get that wrong?
I don't know.
That's what I've heard.
Yeah. Okay. Good. Yeah, but I think nevertheless we should involve him. But I think, I mean, I also can talk again to Gastone and Javier. I mean, they know the paper, and I think the algorithm is published. I think that should not be a huge problem to redo the approach like he did. Yeah, but that's a good suggestion. Yeah. Good. Okay. But now, Maria.
Hello. So good afternoon again. I would like to ask Alan whether he's going to show my slides, or I can share the screen. Okay. Do you see it?
Yes.
Yeah.
Yes.
Okay, fine. Okay, so good afternoon. I will give you an update on the APL project, which is unfortunately not so advanced as the general AML project. So these are the participating groups who already gave an agreement on the project. And as you see, there are many groups very well represented. And these were the project goals that we already discussed last year in Florence. So, and I think that in this setting again, like in others, in APL, we have the advantage of being able to compare some of these issues in the ATRA-ATO area as compared to the ATRA chemotherapy era. We discussed this in the smaller APL group, whether we should keep the ATRA chemotherapy data in the database, which are obviously pretty old, especially for low to intermediate risk APL, while in high risk APL, we're still using ATRA chemotherapy as a standard. And as you know, we have the Apollo project for to compare results in the ATRA-ATO setting. And the goals are the characteristics of diagnosis, de novo versus therapy-related APL, the type of breakpoint. We know that BCR3 has been indicated as a negative predictive factor in APL in the chemotherapy era, FLIT-ITD, the differentiation syndrome and the role of prophylaxis, the different approaches as a, comparing the different approaches, the role of maintenance therapy, which we know is more or less is only important in the chemotherapy setting and higher risk APL, CNS disease, prophylaxis, management, characteristics of elderly patients, and what I think is relevant is also late complications of APL treatments, such as secondary myelodysplastic syndrome or secondary malignancies, and also secondary late effects of therapy, for example, neurological side effects or cardiotoxicity, for instance, in the ATO era. Although I'm not sure whether our database, including this late follow-up or how long the follow-up will be for most of patients, because as we know, some of them are lost to follow-up and considered cured. We have already shown this slide with the number of cases expected. If everything goes well, we expect to have at least 4,800 patients deriving from the different databases and groups. Some of the groups didn't give an estimate yet of how many patients they have. So this number might increase a lot, and this would be really, really a nice group where we could also try to answer some more specific questions. And what we have done is to identify the variables which are essential to enter patients in the database, and these are indicated here in red. And then there are some additional variables that can or cannot be available. And so, I mean, these have been now accepted or at least entered in the system. And now we are requiring the data dictionary, and and the coordination center is also forwarding the data sharing agreement. For most of the groups, data sharing agreement is available for AML, and so we only need an amendment for the APL project. So this could go very quickly. Of course, if for all the colleagues who are present today, I would like to ask them to try to send the list of available variables so that we can really go on and see what we have, and also to send to the coordination office the number of the patients that they think that can be entered into the APL DB. So this is the roadmap. So we really hope to start collecting the data now. I mean, this has taken 1 year to get to the point, and we would like to make a smaller APL project meeting involving all the leaders of the group, but also others who are interested in the project, and try to define some sub-projects in the first quarter of 2021, and try to do some statistical analysis and entering of the data. I mean, Q4 21 is a little pessimistic, but maybe Q2 2021 would be nice to do some first analysis and maybe send an abstract to the ASH meeting next year. As you know, as you see, we are a little backwards yet, but I really hope that if we have sub-meetings, smaller meetings with the group leaders and with other younger, hopefully younger investigators interested in the project, that we can proceed a little bit quicker with APL as well. And now I'm happy to take questions or suggestions from you.
Yeah, thank you very much, Maria Teresa. I think this is a very good project. I think we have to first work that we convince people to provide the data, right?
Yes.
But I think now, as we have I think the process is very well established. I think most of us should have signed the paperwork already for the first data set. So I think if we have a second one to submit to Harmony, that should be not a problem with the institution anymore. And then I think once we have the data, I think the analysis should be also straightforward. Can we have again a look at the overview of people who are going to contribute? Perhaps we can identify some additional people.
Yes. Okay, so this is the original list with the contact people. Of course, this is open to, I mean, I've added some groups last year at the Florence meeting. We have had some proposals from some new groups and also from the US intergroup. This was at ASH last year, but I haven't been able to hear, so maybe the coordination office didn't have any news because they were to sign agreements also with the US institutions to be able to maybe, I mean, also some other European groups could have data. Unfortunately, I think there is not much molecular data on APL. I don't know whether they were included in your AML, in the AML database, the molecular data on APL, or are those only AMLs?
For us, it's mainly AML. I think with many of the studies, APL was excluded, right?
Yes, yes. Just wondering. Yeah.
But of course, we have many APL cases, but then very often they were not as comprehensively characterized using larger panel sequencing but on the other hand, I mean, for a project like this one also could approach people. I mean, panels are not that expensive anymore, and one easily could also do 200 cases and panel sequence them if DNA is still available. Yeah.
Although, I mean, I must say that by panel sequencing, APL do generally have a lower number of mutations compared to AML. But for example, ARID1A mutations have been described in APL who were resistant to ATO as a mechanism of response. So, I mean, for those cases who are unresponsive to our relapse after ATO, a larger genomic approach could help to identify the markers of resistance. And this could be really an occasion to identify those patients relapsing after ATO treatment and collect them and maybe analyze them using genome WES approach or something like this. And this could be really also biologically very, very interesting.
So that means that you, like at the beginning, that also would be helpful to put only the clinical information together of the cases, to have a large database and cohort, and then say, okay, we have identified, but then the problem would be one it's a little bit with this de facto anonymization, we cannot really go back to an individual case, right?
Oh, okay. But could we
but what you could do is then say, okay, we have identified 5 ATO or patients with relapse after arsenic trioxide. 5 came from your cohort. Could you look into these cases and see whether you still would have DNA available? And then we collect these cases and perform a whole exome sequencing study, for example. Yeah.
That would be a nice, I mean, we could really go back to the groups and ask them for the samples. Once we see we have enough patients with these characteristics, and these could
that it makes sense to do a, yeah.
Another one could be, for example, to characterize patients who have extramedullary relapse of APL. Those are known and do occur rarely, and maybe characterize those APL from a genomic point of view, whether they have different characteristics from the general, from those who just go into remission and never relapse. Also, extramedullary relapse would be a very interesting project to, also to approach from a molecular point of view.
Mm-hmm. Okay, but then, I mean, that also could be something to approach the respective groups to at least ask to whether they would be willing to share what they have for now. I mean, clinical information then already would be helpful, right? But we would have to make sure that the parameters are available that you would be interested in.
Yeah. So I mean, in the list of variables, those parameters are there, and so I really encourage everybody who's participating to this meeting today to actively try to push the project. And I think that if we do it together, I think this is a disease model, a rare type of AML where we could get some answers, some further answers. I know that it is cured in 90% of cases, but we can still try to answer to some interesting question, to change maybe our ideas on how we prevent differentiation syndrome, which is an issue, for example, in high-risk APL treated with arsenic trioxide. So I think, there are still some relevant questions that we can answer, and with the help of everybody.
Mm-hmm.
Okay, so I agreed with the Harmony Coordination Office that we will send a doodle to the people listed, to the groups listed, and to others who are interested in the project to do a smaller APL meeting, web meeting, to really, try to define some sub-projects and the people who can be responsible for the sub-projects. But as you said, the first thing is that we have enough clinical data.
Mm-hmm. Okay.
Okay.
Okay, now that sounds like a straightforward plan and doable. Very good. Because I think the clinical data, if we approach those people, we should get together quickly. And then we can go from there and initiate some molecular-driven studies.
Okay, great.
Okay, very good. Okay, then I think we have talked a lot now. I think we have a more concrete plan. For me, it's still a little vague, but I have to. So I think I will, not for your project, but for the other one, I think I will talk to the data analysis people, and then we will approach the people who have expressed their interest today to participate. And then we, I think, should first work hard on this gene-gene interaction recapitulation of the Gaston paper project, and and then go for new questions from there. I think that's, and then we can assign individual people to the respective small sub-projects and have them drive them. I think that makes sense. I think some of you might have to leave. Sorry, who wanted to? Peter?
Yeah, I just want to say one thing. So similar to what was now discussed about APL, you could imagine that you could do more or less the same thing in AML. If you have an interest in a certain subtype of AML, then you may want to approach people from the other centers and do a more in-depth, like a whole genome or whatever analysis on a subset of cases.
Mm-hmm.
Because, so as we said before, so if you only do the gene panel analysis now, in a way it's a little bit confirmatory or what has already been done. But then at some point you may be able to identify a subset of cases that would be of interest to do a more broader genome-wide analysis, or so similar as what has just been discussed about the APL. I think. I mean, I could imagine that.
I mean, I guess for many of the questions, or some of the questions, many of us might already have the data, and we also could put them together. But of course, we have started to put data together for Harmony, which everyone likes to share.
Absolutely.
And that is, but that's a good point. Good. Thank
Thanks,
you very
folks.
much to all of you.
Lars.
Thanks, Lars.